Welcome to this video on post-translational modifications in proteins. In this video, we will learn how proteins, once synthesized, will undergo important chemical changes that will enable them to perform specific roles inside our cells. These changes are called post-translational modifications, or in short, we can say PTMs, and they are essential to life. So let's begin with what are post-translational modification. After a protein is made through the process of translation, it doesn't always function right away. So it often requires further chemical changes, and these modifications refine the protein's shape, and it controls its activity and decide where it will go inside the cell. There are over 200 known types of PTMs, and they are found in nearly every organisms. So, why are PTMs important? Post-translational modifications are crucial for several reasons. They act like switches, turning proteins on or off, and they guide proteins to their correct locations inside the cell. And PTMs also help proteins interact with other molecules and protect them from being destroyed too early. So, without these modifications, many proteins would be useless or even harmful. So, now let's see the types of PTMs. Let's now take a closer look at the main types of post-translational modifications. The most common ones include phosphorylation, glycosylation, and ubiquitination, methylation, acetylation, and lipidation. Each of these types adds a different chemical group to the protein, and each has unique effects on the protein's function and fate. Let's begin with phosphorylation. This process, as the name says, it will add a phosphate group to certain amino acids in the protein, and usually it's like serine, threonine, or tyrosine. These are the amino acids to which the phosphate group is usually added. And phosphorylation acts like an on-off switch for enzymes and signaling proteins. It is especially common in pathways that control like cell growth, metabolism, and communication. Next is glycosylation, where sugar chains are added to proteins. This modification is important for proper folding and also to maintain stability in many proteins, particularly in those that are secreted from the cell or embedded in the membrane. Glycosylation also plays a key role in immune system recognition, allowing our body to distinguish its own cells from the foreign invaders. So, in the immune cell recognition, that is from the self cells, to the foreign cells or foreign substances, how does the immune cells find that which cell is self cell or a foreign one? So one of the important property by which the immune cells recognize the self and foreign cells or invaders is through the presence of sugar chains. That is, this glycosylation plays a key role in recognizing the self cells so that it can distinguish the own cells from the foreign invaders. The other main PTM is the ubiquitination. Ubiquitination works a little differently. Here, a small protein called ubiquitin is attached to other proteins. This will mark them for destruction by cells recycling center called the proteasome. So this process will ensure that the damaged, misfolded, or surplus proteins are efficiently removed, thus maintaining the cell's health. So, ubiquitination is basically for removing the proteins which are unwanted or which is in high quantity, which is not required by the cell. So, ubiquitin, which is the protein, is added to the particular complex, and then the substrate is destroyed by the cell's recycling center, which is the proteasome. So this is the function of ubiquitination. Methylation and acetylation. These two modifications often happen on histone proteins. This histone proteins we have seen in the package of DNA inside the nucleus. So by modifying histones, these PTMs can control how tightly or loosely the DNA is wrapped. This in turn regulates which genes are turned on or off. That is the key part of what we call epigenetic control. So turning on or off a gene is carried out by uh, these processes like methylation and acetylation. We have seen that phosphorylation turns on or off the enzymes, that is, the enzymes which play key roles in the cellular functions. So similarly, methylation and acetylation will work on the gene, that is, it is working on the histones, which is the DNA packaging proteins, which tightly packs the DNA into the chromosomes we have seen in the DNA structure. With these modifications, the gene will turn on, off, so that the gene expression is controlled, and this is responsible for epigenetic regulation. And the other process is lipidation. Lipidation involves the addition of lipid or fat-like groups to a protein. This helps anchor the protein to cellular membranes. Many signaling proteins, especially those involved in growth and communication, rely on lipidation to function properly by positioning them in the right place at the right time. So these are all the types of post-translational modifications in proteins. They help in proper functioning and positioning of the proteins. But when PTMs go wrong, problems arise. Abnormal phosphorylation can lead to cancer, while faulty ubiquitination is linked to neurodegenerative diseases like Parkinson's, Alzheimer's. Defects in glycosylation can cause autoimmune disorders. That's why scientists are actively studying PTMs as potential targets for new therapies. So, post-translational modifications are chemical changes that transform simple protein chains into complex and functional molecules. They regulate protein activity, stability, location, and also their lifespan. So, understanding these modifications is critical not only in basic biology but also in medical research, where they are increasingly targeted in drug development. Thank you for watching, and I hope this session has deepened your knowledge for the hidden chemistry that powers life. And this is how the post-translational modifications occur, and these are all the types. These are necessary for proper functioning of the proteins, which are building blocks of life. See you in the next video.